Hello, my name is Jimmy Kim, and I'm one of the many co-founders of SparkLabs. And we want to welcome you to our panel, our webinar on rare genomics. No, not rare genomics and its impact for the future. And the official name is the growing impact of genomics. So I want to welcome two very distinguished people in their field. And I want to first, ladies first, and Caroline, I want you to just, I don't want to take away anything, so I want you to kind of just introduce yourself, what you're doing right now, how'd you get there right now.
Sure. So I'm the scientific director in the Department of Genetics at Yale. I've been here about 2 years now, and my background is in genetics and specifically stem cells and regenerative medicine. Part of my role in the department here is to think strategically about where genetics is going as a field and how Yale is going to stay at the forefront of that and be leading that effort, particularly with regard to the specific strengths and opportunities that we have in our department. Because genetics is a very broad field, I think about what is it that we have and what can we bring, and how can we leverage the amazing talent that we have in our department to keep pushing the envelope on genetics. Another part of my role is to work very specifically with individual faculty. So we have about 35 different faculty in the department, everything spanning from clinical genetics to very basic genetics to new sequencing technologies and all sorts of different things going on, cancer, neuro, whatever. And to work particularly with junior professors about how to kind of shape their research program and really leverage their own individual strengths and make sure that they're tracking where genetics as a field is going, and so they're going to continue to have that sustained growth through their career. So it's lots of different areas of genetics, all biomedical, but really thinking about where the field is going and where those opportunities are in the future.
Great. You had a pretty interesting traveling worldwide thing. Can you explain what continents that you lived in? And,
Sure. Yeah, absolutely. So if you're lucky, you might get all three accents coming out in this. I was born in Australia, and from Australia, I studied at the University of Queensland at the IMB, and then I moved and started a postdoc in New York in a stem cell lab there. And then following that, I went to the United Kingdom, where I worked as an editor for a scientific journal for 5 years, really focusing on stem cells and regeneration. And then after that, came here to Yale in New Haven into the genetics department. So it's been all around the globe, and who knows where it'll end up. I hope here, but.
Well, maybe Asia. That's one of the
maybe that's the missing piece. Yeah, maybe that would be fun. I love Asia. I've been to Asia many times. Many Australians have, and it's fabulous. So yeah, that would be cool.
Great. Well, Jimmy, I like your name, Jimmy. Why don't you kind of give your background, where you are, and everything right now. I'll ask some other prompting questions also.
Great. So Caroline actually overlap at different times, but I did my undergraduate training at Yale, where I worked a lot with the genetics department. At that time, elucidating the genomes of the first 20 organisms, first ever sequence before we even had the human genome sequence, doing sort of comparative genomics, phylogenetics there. After my work there, I did my MD PhD at Johns Hopkins, working on cancer genomics. There, looking at, we were working with Bert Bolestein, were the first to really elucidate after the human genome was sequenced, what the cancer genome, looking at the genomes of individuals' cancer and how that compares to the normal, and what are the mutations that potentially cause the cancers, and thinking about drugs or diagnostics for that.
Right.
After my MD PhD, been mostly in academia at Washington St. Louis and at the NCI, and then had some sort of different industry where I was at Natera developing a cancer monitoring test, and then currently I'm the chief scientific officer in a venture-backed startup called Freenome. We are interested in the early detection of colorectal cancer using a blood test. There's millions of people in the world that we are not being screened for colorectal cancer, and because of the methods that we use are very invasive, like colonoscopy or stool. People don't want to do it. So we're developing a blood test to do that. So that's sort of my day job. And then my passion work that I like to call is I run a nonprofit called the Rare Genomics Institute, really stemming from part of my graduate work where some of my genomics experience and also meeting families with rare diseases that could not be understood what it was. So we started this institute about 10 years ago, and then have really sort of expanded now to, a staff of about 200 people in 7 countries, really helping and looking at ways to initially help diagnose these patients with rare diseases, and then figure out what downstream research is needed. And now even thinking about phenotyping and medical records and doing clinical trials to think about therapeutics for these patients. So those are my 2 main hats now, Freenome and Rare Genomics.
Great. I mean, I guess we kind of covered your history, kind of, right? So in your, I mean, Jimmy, you kind of mentioned this right now, but Caroline, in your, you have 35 junior, PhDs, and you're working with professors right now. I mean, what are some of the interests right now? Like I said, that you're, as a department or as your individual, I mean, where are you really like reading a lot of papers on and, following, following some professor? What are some of the fields right now that you're really, keen on these days, because kind of you think that's going to have a great impact on?
Yeah. I think one of the things from a very basic research perspective is really understanding now the next frontier of genomics is really to try to understand what all the non-coding parts of the genome are doing. And I think that's something that's really exciting from a basic genetics perspective, and how all those feedback mechanisms are going on in a cell to try to regulate our gene expression. Now that's a very basic kind of fundamental genetic question. I think some of the things that are probably a little more accessible that are happening here as well in our more applied, our professors that work on more applied genomics are, things, for example, like preventative medicine. So we have a project called the Generations Project, which is screening 100,000 healthy individuals to try to understand what's in their genetics that keeps them healthy, and what's in their genetics that might in the future lead to disease. And this is really kind of based around this idea of, can we use a person's genetics to try to assess health outcomes? And if we can, then can we start to intervene and to do like targeted screening or enhanced monitoring to try to zero in on those patients that are more at risk of certain diseases, whether it's cancer or heart disease. And so this is a study that's being run out of our Yale Center for Genomic Health. And it's really cool because what we can do is link it with all the EHR records. So we have all of those on those patients, and then we can start to do, all of those data are available to our researchers, and we can start to do really cool, interesting studies looking at not just the genetics of the patients, but also all those, family history, medical history, the environment, all sorts of different studies, including like the public health implications, policy. There's all sorts of things that you can do with those data. So that's one thing I think that I'm really excited about that's happening at the moment. Another would be AI. I think that we've got a really wonderful faculty, Smita Krishnaswamy, who is a computer scientist, but she's appointed in the genetics department because she is really at the absolute forefront of combining multimodal data sets. So not just genetics, but single cell RNA-seq with ATAC-seq. These are basically different omics technologies to try to extract really meaningful data that is not easy for a human being to extract. So that's machine learning, basically. She's at the forefront of machine learning to try to harness these massive data sets that are getting produced now all over the place. So that's kind of a couple of, I would say, the exciting things that are coming out.
Very interesting. I was studying, we used to think that there's non-coding DNA used to be garbage, which is kind of like, really? And then,
exactly.
And then
I mean, we were all taught it was junk DNA, and the fact is that there are now lots of reports, many, it's very well established that it is not junk DNA. And these are in fact regulatory regions that are controlling how and when genes are expressed. And when there's a mutation or when something goes wrong, that definitely can have a disease phenotype. But it's just really hard to understand, like, all the different variation that can occur in those regions. Like, is it functionally significant or not? And I think that's where we're at, is trying to understand at what point is sequence variation functionally important and at what point does it not matter. So that's kind of, from a basic research perspective, one of the things that we're really interested in.
Yeah. So Jimmy, let's talk about your passion project. I mean, I really think it was 10 years ago when everybody was like, when the genomics precision medicine was really, kind of like, hey, right now. But I really think that, starting out from a nonprofit and, where it is right now and, right now, I mean, kind of shared before right now, but can you just, there's some great stories, that I know, but I'm prompting Jimmy about the things that happens through rare genomics and everything right now, and where you guys are going. It will be very interesting to hear.
Yeah, I mean, the story started off right during my PhD. We were among the first to sequence the cancer genome, and I was really excited and proud to be part of those efforts. And then during my medical training, we ran into, I met a small kid with a rare disease that was undiagnosed disease. And me being at Hopkins and being this sort of bright-eyed medical student, saw this family come in and like, and you do all the questioning, and then you go with the doctor to the back, the attending, and ask like, so have you now figured this out, Dr. House, right? Like thinking that it's amazing. And then and this particular patient came in and he's like, well, this family has been to the top places at Mayo and Harvard, and frankly, I don't know what additional tests I would do. And that they were just like, whoa, that this family didn't have an answer. And I remember still very clearly when that doctor told the family that, we don't have any additional testing that we could think of. And seeing that whole family sort of, it was a whole family that came, the whole tribe, if you will, walking down that hallway and thinking that there must be something we can do if there was a thought that this disease was a genetic disease. And that's when we sort of embarked on this sort of early ways of thinking about sequencing people's genomes for rare diseases and being able to solve that. And we started by these sort of crowdfunded grassroots efforts to help patients one at a time. And we've and along the way, we've discovered new diseases. And this, the cool thing is, and now this is an entire field. There's big projects now at Yale and at Johns Hopkins sort of doing this. But when we first started, we were really excited because this was research that was not yet being done and was funded by like aunts and uncles who were giving $5 there, $10 there, and was using excess capacity at top research centers at that time that, because it was expensive, and with volunteers at these academic centers who wanted to make a difference. And in these 10 years, I'm so glad to say that, a lot of this is already in clinical practice. So these kids oftentimes would now, the things that was sort of a pipe dream for us to do. And then one of our, the main producers of this equipment, Illumina, actually does a lot of this pro bono for many of our patients now. And there's still legislation that we're pushing past forward to get this for everybody, but there's still families who really need it, and we help those. So we're excited that, in this 10 years, this was a very, very hard to solve problem, which is now, for some patients, standard, almost standard of care but then the question is, once you figure out what the gene is, what do you do about it? What does the gene do? What are the functional analysis? And we're helping, we work with companies that will create mouse models for these families or do CRISPR and then sometimes you do that, that's not enough. So we're working with groups of families to figure out, for a whole group of these families, what's in common that they have these changes? Are there natural history that we should understand? And even recently, we've launched projects into wearables to figure out, are there ways that we can understand the phenotype of these patients to be able to really better characterize the genotype as well. So I think, again, the science that we do pales in comparison to the multi-billion-dollar projects of many of the academic centers. But what is exciting is for a lot of these families who otherwise are not being helped through these efforts, they come to us and we then custom design research projects to be able to help them. And then through these different sort of efforts, sometimes through crowdfunding, sometimes through pro bono, we're pushing forth. And what's exciting is many of these projects then receive downstream government funding and birth new research projects looking at diseases that are otherwise not studied. And the cool thing is I get to meet a lot of these families. I've been to birthday parties, 18th birthday parties. I get to get invited to a lot of these sort of celebrations and just to see that impact. And for a lot of the scientists who volunteer with us, many of them in basic science who actually don't get to see their work impact humans, this is very meaningful to them as well. So we're very privileged to bring the scientists to the patients and bring the patients to the scientists.
Well, I guess Jimmy kind of opened the two of the C's I was going to talk about today, COVID and CRISPR. And then I think that, CRISPR, yeah, there's ethical stuff and and that we'll touch upon, I guess, a bit later. But, Carolyn, what's your, like you said, in the basic science part and everything right now? CRISPR, of course, it's getting a lot more hype than a lot of other technology right now. But your study with stem cells before and everything right now. I mean, what do you want to share your views on CRISPR and where you guys are thinking of going with it?
Yeah, I mean, CRISPR has changed so much. It's been wonderful for all sorts of different things. For gene therapy is the obvious application that everybody thinks of, and we have actually. You must know him, Jimmy. Do you know Monkol Lek?
I actually don't.
You don't? So Monkol.
Yeah.
So let me talk about Monkol Lek for a second, because he's also Australian. So Monkol Lek is one of our faculty here. He actually he was a computer scientist who was then diagnosed with muscular dystrophy, and they couldn't find his gene. They couldn't find the mutation, and so he stopped his job with IBM that he had worked for his whole life and went back.
Oh, okay, I know. Okay, now I know.
Yeah, you know Monkol. Yeah, exactly. So he went back and studied and did his PhD in human genomics. He found the mutation. He went to Broad. He led the many very important studies there, was part of the CMG there. And then we recruited him here as a junior faculty, where he is now embarking on the very first gene therapy CRISPR trial in muscular dystrophy. So he's in discussion with the FDA now, and that's part of Cure Rare Disease. That's a partnership with Cure Rare Disease. You must know them. So, getting back to your question, Jimmy, CRISPR, we all think about gene therapy. It's the obvious application, and it's something that we certainly are also working towards. I think what's probably not appreciated so much by the public is that CRISPR has been absolutely just a game changer for a lot of model organism studies. It now means that you can make mutants to study diseases. You can do these massive screens for, you can put a mutation in every single gene in a zebrafish, feed it through a big machine that looks, takes a photo, completely automated, of every fish that passes through, and then AI is looking to see if there is any, skeletal abnormality or a neural abnormality. It's so high throughput now. It's a massive production line, and I think CRISPR has had a massive role to play in that. And there's been a renaissance in developmental biology in model organisms, because now we can really start to look in vivo in high throughput, and that is very, very powerful.
Great. Yeah, I know Salk and all the people are using zebrafish, and I think CRISPR definitely has definitely sped that up. And it kind of reminds me, when I was doing my PhD, when polymerase PCR first came out, and I haven't seen anything this so-called pervasive or game changing in the field either. You know, right
Yeah,
now.
it's a big game. It's been a huge game changer, I think. And you know, technology only tends to get better. So
Yeah.
you know, it's
Yeah.
definitely.
Well, yeah. Well, you know, ethics is not something that we can talk about right now, but we'll catch up a little later. But I want to talk about Jimmy too. I mean, being at the rare genomics field, and right now, and your stuff with Freenome. I mean, CRISPR. I mean, how yeah, how has that changed your stuff and your view, and what are some applications that you guys are using it right now?
Yeah, what's exciting about CRISPR is it's not right. There's so many new enzymes that people are also finding for different types of enzymes for CRISPR, and then different of those enzymes open up new opportunities for research and fidelity for that. So I think about CRISPR initially, we're just there's sort of very small set, but now there's sort of large searches of other CRISPR enzymes, and then CRISPR initially, and now we're thinking beyond gene therapy and even like diagnostics. There's multiple diagnostic companies that are now using CRISPR as a reporting mechanism. Some of them, I know you're going to talk about COVID, using it for COVID. So to develop almost sort of point of care devices. Think about sort of similar to like a pregnancy test that you can put CRISPR, and CRISPR no longer as a tool to actually edit the genetics, but as a reporting tool as well. Think about, I know you mentioned PCR, the new ways of it's similar to that, to read a specific sequence. And what happens is CRISPR sort of chews up different areas, but if you chew up a inhibitor to a reporter, then it can be used for a diagnostic. So I think it's one of those things, like, thinking about computers, you think about multimedia. People are like, whoa, multimedia, that's amazing, but it's going to be so pervasive in the world, or cell phones, that it's such a fundamental technology that it will just be part of everything that we do, and to be used in those aspects. So I think, not only sort of research tools as well as diagnostics, but you mentioned ethics is like, right, with all these tools, there's going to be unintended consequences and problems there as well, in the same way that cell phones causing potentially problems with as well. So I think it's going to be such a pervasive tool that, again, like you mentioned, PCR, like everybody, and it's so easy to use. And that's the beauty of it. It's so programmable. The number of uses it's going to be used for, it's sort of thinking about like, what can you use a computer for, right? Or a cell phone for. It's going to be so pervasive that it's going to underlie everything that we do within healthcare.
So I mean, we're definitely. I just want to. Caroline talked about this 2 days ago, in the sense that of apologenics and the skew thing right now. I mean, we're definitely a lot of people are talking about, hey, precision medicine. It's kind of like, I sometimes feel like the media gets ahead of us a lot of times, saying that we're saying that we're almost there. Precision medicine. There are people who are very pessimistic, saying that we still have a long way to go, and the people who are like, it's already here. So I get two responses when people get like a rare disease, and right now, oh, don't worry about it. There are people who are like, oh, I'm very fair right now, and then so what are some of the from the field we talked about right now, what are some of the areas still, as much as we've gone so far, some of the places that we still need to do the grunt work on right now? Caroline?
Yeah, one of the major gaps in genetics is the fact that almost all of the studies on human populations are based on people of European descent. And this has major implications for how we interpret genetics and how we apply that interpretation to patient populations. And so what we find is that a lot of the genes that get reported as being involved in a certain disease or predicting risk, or more predicting risk, I would say, actually, but a lot of the genetics that underlie some of the things that have been reported don't actually hold up when they're applied to patients and populations of different ethnicities. And we see this particularly with things like polygenic risk scores. And so there really needs to be a lot greater depth and breadth across our sequencing studies in order to be able to properly interpret what variation actually means, because there are large tracts of the genome that are specific, that that you can look at, that are specific to different ethnicities, and what variation means. We see phenotypic variation, and of course that's going to be reflected in our genomes. And so we need to understand and kind of dig into that and get to the real biology of what's going on before we can start to apply findings that have come from predominantly European populations to the rest of the human population. So that's one area I think that we still have a ways to go. Particularly with when we start to talk about risk.
Yeah. Jimmy, what's another area, or do you get some areas that think, hey, to say, hey, this is still, yes, we're very happy that we're getting a lot of spotlight in, precision medicine, right? But hey, we still have here are some of the gaps I want, that we should know about, and everything right now, or where research is still a little bit lacking or progress is lacking on.
Yeah, so I talked to my colleagues in other sciences like physics and chemistry and math, where they can almost predict with certainty. We can land a rover on Mars with absolute certainty, and then we talk about biology. Like we can read a person's genome, but we have no idea of what most of those genomes work. And I think it's pretty amazing that we can read the genome for the first time, and these recently. But human biology is so beautifully complex that every cell in your body has a different sort of RNA program there, right? And a different history, and they interact in different ways. And then proteins are even more complex, and we can't assess all the proteins even in one cell yet. So yes, I think we've come a long ways since we're doing very basic, just sort of measuring size of skulls, but we're just getting started. We are just getting started. Like the human genome is the easiest part to understand of the human. It's the part that mostly doesn't change. It changes a little bit, with cancer and hereditary, but and it's mostly the same within your body. But that's the first part of the central dogma, right? RNA is the right, and the central dogma, part of it holds and part of it doesn't, right? You think of RNA, we know very, very little about different cells, and RNA changes over time. So DNA is mostly static, right? Unless you get mutations, but RNA changes over time. And then on a protein, we don't even know what the proteins are in every single cell. Our proteomics is still, there's a lot to be learned there. And then there's post-translational modification to proteins. So I think we are just starting the journey of understanding even the DNA. We don't know how it changes, RNA, protein changes through time and space. And there's some, where, the field is starting to look at single cell work, but even that is just getting started. And we don't know how different parts of the different cells interact with each other. So I think we're just at the start of the journey, but it's a very, very exciting start. And sometimes I would like to joke that we really don't know what any one protein does in the body, because it works as it interacts with different proteins at different times in different ways. So there's some proteins we study very, very well, like the big cancer proteins, but unless their effects are very, very significant, will be. So I think in biology, we're good at finding, things that are very, very single cause, like the Mendelian diseases, right? Like cystic fibrosis, or if the causes are very, very clear cut, right? But a lot of things are not clear cut. It's sort of like weather. So when it's more complex, like you mentioned, polygenic diseases, we're just the beginning of sort of understanding that complexity. So I think we're at the very, very beginning of a very exciting voyage.
So I mean, this is kind of relates back to kind of what you said about the non-coding genes, right? Protein coding genes, right? We're trying to find out that, and we're also to find out like a transcription, translation, post-translation. There's a whole thing from DNA that we're are going to go up, and then the whole thing of studying healthy. I think that's a very interesting thing, right? You kind of wonder, why is that guy so smart, or why is this guy so healthy?
Yeah, how come this person can eat whatever they want and they perfectly healthy? How come? How come people live to 120? What is that?
Yeah.
That's really interesting as well. Studying not just disease but health.
Yeah.
That's really interesting.
That's definitely an area I think that's going to move forward. Now let's talk about the other C. Right now, we live in a different world, right? We're all at home, or Caroline had to drive to her office to make sure she had a good connection and everything right now. But COVID, I've seen, you see grants. It seems like everybody that I know who used to study cancer now is doing COVID stuff. Right now, which is not, I see the urgency, but I am kind of concerned also, in a sense that, there's a lot, people have to follow the research money and grants and investment where it's going right now. And definitely right now, how has COVID, changed your perspective or agendas, in your respective kind of, entities that you are in, and even, in person,
Yeah. Jimmy, you wanna
Yeah, Jimmy,
Ladies
Jimmy.
first. I thought, yeah, happy. Yeah.
Sure, yeah. So I want to start by saying, I've witnessed Yale's response to the pandemic, and I think it has been exemplary. From the absolute get-go, it was all safety first, and then it was, okay, what are we going to do about this? And everybody pulled together across the med school and across the university to tackle this. And I think what we see now is, at least, what the world is seeing now is one of the fruits of those labor in the saliva direct test kit that just got FDA emergency authorization, and is basically, it's going to be instrumental in getting everybody safely back. Our undergrads are coming to campus. It's obviously modified, but they are coming to campus, and schools are returning now in the fall. And so, what they've been able to do by developing this saliva-based test instead of the nasal swab has been fantastic. And, it really took an effort all across different aspects of Yale. And so I think that, the ability to produce something like that from everybody's collective talent, and to not commercialize it, but to make it freely available to the public, and based on, very simple lab reagents. It's $10 a pop. It's really something. It's a gift to everybody to try to beat this thing, as soon as possible. And I think that, yes, I felt it too. Like I'm heavily involved in grant writing, and I felt immediately the pull of COVID and all the funding that gets diverted there, and everybody's really invested in trying to solve this and to beat it. But it's for a reason. It's because it's just an unprecedented toll on our lives, on our lifestyles, on everything. On our revenues, just everything. It's affecting everything. And so what it shows is what we can do when we pull together, I think. So, yes, it has diverted a lot of funds, but rightly so, I think, at this point, because, we have to get a handle on this thing.
So yeah, I was very, I saw the news of the Yale saliva kit and $10 a pop. It's definitely, it just shows that, when people multidisciplinary solve a problem.
Yeah, absolutely. It probably, it sounds so neat with the way the media parcels it up, but the people here have just, I think they've gone 5 months without sleep. It's just phenomenal the way that people have pulled together, the deans and the different schools across Yale. That came out of the School of Public Health, but it's all, there's so many people involved in that kind of thing. And I think it's getting used by the NBA now, the National Basketball Association or something, I heard. So it's really having a wide reach, and I hope that labs across the world can use it. It's for anyone out there, the preprint is up on medRxiv, and anybody can do it. So
great. Jimmy?
Yeah, I think it brings out, I mean, with all crises, sort of the best and worst that we see. It sort of highlights, right? I think let's talk about the best first is right for us to as a humanity to develop different diagnostics so quickly, right? We got the genome sequenced really quickly. And I know, even because of the Asian countries has been responding to SARS and others, some of the precautions. So I think, yes, it could always be faster, but the fact that we're able to get diagnostics out so quickly, and it always could be faster, but as a species, for that technology to be made available, antibody tests, and across the world, and then now sort of possible therapies, right? Different areas, vaccines and some therapies. And it is in record time. Like if you think about any other disease that, again, it could be faster, but within, this sort of year to be able to do that, to mobilize, to do that. So this is the great part. I know it could always be faster, but I think for the scientific community to come together for all these different aspects, it's been great. The sad parts are there are such simple things like public health prescriptions that that it can be done that sometimes aren't not done, and it sort of sometimes reckons to sort of me fighting cancer and thinking about smoking, right? Like the cure for lung cancer is to stop smoking, and there's such simple things. If people stop smoking, that probably will save more lives than me developing a lung cancer diagnostic test. So there are simple things that sometimes like we feel like, wow, scientists are going sort of over, out of their way to develop them, but there's some simple measures that potentially. So I think there's sort of the positive and negative sides, and it's exciting parts of this that really sort of highlights there. And I do think that, that the fact that, we were able to make scientific progress so quickly, still worth, from my optimistic scientist hat, that's something sort of worth celebrating, but still, we need the whole public's buy-in and sort of for actions, and you can see that across different countries there for that to happen. So ultimately, doctors can and scientists can create the most amazing things, but ultimately it still needs sort of public adoption. So I think COVID-19 shows a lot of that, because there are very simple things that we can do and very complex things we can do, and it shows both sides there.
I think it's like you said, I like that analogy of you say, hey, I got a cure for lung cancer, but you gotta stop smoking right now. I know. I mean, definitely. Like I said, when the Yale staff labor 5 months and everything right now, not just because you can get tested easily doesn't mean, hopefully, no masks right now. No, I think like I said, you can only well, like I said, it's very, very, very encouraging. It just shows that when you one thing, multidisciplinary, when you have focus,
Yeah.
people can get to. I think that is definitely a celebration of the collaboration that can happen moving forward.
I hope it really shows the public how important it is to keep investing in science and in health. It's very easy to cut the budget when times are good, but you need to keep it well funded. It's a machine. So
yeah. So that was my next question. Right now. I mean, to biological sciences. Right now. I mean, when you're there, I mean, it is the basic sciences that fuel, I guess, the applications and everything else, else right now. And Caroline, you're, like I said before this, you had a little angel investment, kind of like mindset. Also, you worked at an angel investment. You personally saw what's going on, right now. So, but. And you're writing grants right now. What are some of the so-called cutting-edge stuff in biological sciences that is just starting, that is just kind of percolating right now, or it's slowly that it's getting your attention and other people's attention also in the biological sciences part?
yeah, that's a great question. I think that there are opportunities emerging in certainly in omics, in multi-omics approaches. So we kind of many of us, I hope, have heard about things like single-cell RNA-seq or single-cell ATAC-seq, and these are, and of course, genomic sequencing. These are applications that allow us to kind of look at what is going on in a cell within a particular framework or parameters. But what's emerging now is basically being able to combine a lot of those techniques in a single fell swoop, and being able to then extract all of that data and to analyze that data. So I think that the the technologies that enable that are at the cutting edge. So combining, for example, in a single experiment, being able to do single cell spatial transcriptomics plus epigenetics plus genetics, you know, all in one go. And any technology that can start to do that, you know, we've got a wonderful, it's actually a physicist faculty. So do we actually have any geneticists? But we've got computer scientists, physicists. We have this wonderful physicist, Stephen Wang, who is developing these types of multiplexing, multimodal omics data, omics technologies. So that's obviously one really exciting area. And I think too, like I said, we still have so much more sequencing to go. So I think there's still, exome sequencing is pretty low cost, but I think other types of sequencing, like whole genome sequencing, that really starts to look at those non-protein coding regions, technologies that can drive that cost down are going to be, would be exciting to me, because I think that there's going to be a lot more of that coming up in the future. And I think AI is obviously a huge one. So, you know, any AI developments that are really looking at medical data, I think the kind of low-hanging fruit at the moment is like MRI data, image-based data, because we already have those kinds of, in theory, those technologies like facial recognition, whatever, pattern recognition and imaging. But then the kind of the bigger challenge is starting to integrate those data with genetics, with electronic health records and natural language processing, and sort of linking it all up into this seamless AI machine learning algorithm that can tell us what's wrong with us at any point in time, even incorporating environmental data. So let's say you're in a city that's really polluted. You know, you get your wearable, it pings you, it tells you your oxygen is doing something. Like it's all possible, but you know, the technology isn't there yet. And so what I think I would be really excited about is some of the AI technologies and companies that are starting to come out that are looking at how to address these problems and how to integrate multiple different data sets. So yeah. They're two areas. I can keep going.
We'll give now Jimmy. You know, he has enough head start to talk about. Jimmy, if you had your hat.
Well, let me just agree first with Caroline, right? Like our company is an AI multi-omics company, Freenome. It's right. So we are based on the Google campus with a big AI department and building. And then the thing with AI is. Machine learning is very different in biology versus in tech. So tech ML is much more what sort of Andrew Ng talks about is decisions you can make in a second, right? And computers can do, right? So image recognition, speech, like all the self-driving car stuff is all stuff that is things a human can do quickly. AI, you know, all the jobs, you know, or some sort of easy document processing, you know, recognition of shapes, those things right now, mostly in tech, that's where ML is a strength. A lot of and then even some of the early problems that ML is tackling right now, I think like Caroline mentioned, like in the other side, hard problems that humans can't solve yet. Hard problems that humans can't solve yet, right? So think about AI, right? Learning chess or Go, and that comparing to people, but then beyond that, right? Solving problems that we're working on early detection of cancer using multi-omics, and we have you know, billions of data points per patient. No person is able to do that, so we need to create new ML infrastructure to be able to detect that. So I think ML in a lot of bio is thinking about transfer learning of different fields, thinking about of methods where, again, it's no longer lots of shallow data, it's some very, very deep data. And ML looks very, very different. As I think a lot of the ML opportunities are sort of flocking into, where big data is and solving it using like CNNs and deep learning, where you can just throw it with big data. I think a lot of the biology ML problems are the other way around, where we need to rethink I think there's still a lot of low-hanging fruit on sort of the deep learning parts, but I think there's a lot of unexplored opportunity outside of deep learning ML in the machine learning areas there. But that being said, data is still very important. So any methods that is digitizing biology, whether it's sequencing, that's the very popular one, right? But the ones that Caroline mentioned, right? Spatial transcriptomics, right? It's going to be all the single cell stuff. Of course, existing proteomics, but anything that digitizes with the electronic medical health records, right? And then I've been saying this again, any phenotypic information, where it's for wearables, measuring your sort of human, what's often called the digital exhaust, data that we just generate in our day-to-day. Ways to sensors, all types of different sensors, from sort of Apple Watch to Google Home and Apple Home, right? So I think there, data generation is now more and more pervasive. And then the key is, how do you take advantage of all this data? Some of it is meant for health, and some of it is just how you carry your cell phone. Will also already tell you your steps, right? Or your speech. There's methods to look at speech for Parkinson's just through a phone call. So I think that the key now is to sort of centralize and capture that data, and think about new ML frameworks to sort of make sense of that data. And then what we do, again, we're biased, but we're interested in early detection and prevention. So don't wait till someone is already very, very sick and stage 4 metastatic cancer to then try to figure out a miracle therapy. We need early intervention. Don't wait till they already need, a triple bypass or a liver transplant. So what are ways that we can do early intervention and early detection of different diseases with existing data? And that's going to be transformative. So I think we're on a precipice of, we're generating lots of data, and we're using so many devices, and we have cell phones in our pockets. We have Google Homes in our rooms. We have smart TVs. We have internet, and now the thing is, how do we now start leveraging that for health? Given there's a lot of privacy questions, there are a lot of sort of research use questions, but I think we're at the precedent. We have enough compute, potentially enough data generation that we can rethink how biology is done.
Definitely. Well, how about this? We're this is one of the I feel like back at home, because this is my major, my master's degree and everything right now. But I was going to open it up to any questions out there. If there's any questions that you'd like to ask Jimmy or Caroline, I will definitely try to relay it, right now. So feel free to add any questions and everything right now. I thought, well, Jimmy, you touched on something that's very dear, that's very keen to me about early detection. Early detection of being, I really think the precision medicine that we're talking about is really about early detection. Right now, of trying to, because I said I was that case when my father had cancer, and there's not, like I said, it was really waiting for the miracle drug, a miracle therapy right now, which we thought that if there was a way to do early detection, right, especially for pancreatic cancer, right now, which is so hard and everything right now. So, let's talk about, Freenome. I mean, how how, where do you, you guys are definitely focused on prevention and interventions. Can you talk about a little bit more on the science or the methodology, the differential methodology behind it. And that's why you joined as chief scientific officer of this company.
Yeah, happy to. So some of my work, so I was part of the team that mapped, amongst other cancers, the pancreatic cancer genome for the first time. And when I was at Johns Hopkins. And we worked with Martin Novak, who's a scientist, a mathematician at Harvard, to knowing the mutations that we detected, what was the evolutionary history of that cancer to that point, specifically in pancreatic cancer, since you also mentioned that. And what we've discovered is, depending on the cancer type, it takes decades, years to decades for those cancers to develop, for pancreatic cancer. Those cancers has been sitting in most of these patients with pancreatic cancer for a long time. And because of where it's situated, it doesn't cause any pain. And only by the time it's symptomatic, it has already sort of very significant sequelae there. So that is the opportunity, that because cancers take such a sort of a progression to that we're detecting it so late, but then it's a slowly progressing disease, really. How can we then find it much earlier, so then it can be, we can intervene? So that's what we're doing at Freenome. We're starting with colorectal cancer because we know that detecting early colorectal cancer, there's a lot of therapeutics and surgery, and people live longer lives. And that often actually is not a given sometimes. Just because you can catch a cancer early doesn't mean that you can intervene. But colorectal, there's been decades of research showing that. So we're doing that first. And then the cool thing is, we're then thinking about other cancers and layering on top of that test. So right now, for example, for ovarian cancer, it's very, very hard to detect. To make a test that's commercializable, it's actually easy to detect, but hard to commercialize because so few women get ovarian cancer. So in order to find that one woman who get ovarian cancer, you need to screen hundreds and thousands. And so for society as a marginal cost, in order for us to find that one woman with ovarian cancer, you're going to have screen a lot of other women, and many of them turn out to be false positives, and they have false positives as problems there too, and it's additional cost, right? We need to spend cost. So, but if we're sort of doing colorectal cancer screening, we can add, for example, ovarian cancer on top of it with marginal cost, right? And for reflex testing, then we can start thinking about when we're detecting multiple cancers in a very cheap way. And then the goal then is to ultimately be able to screen for all cancers through a sort of simple blood test that we're able to do annually. So again, we're starting with colorectal, but we're thinking that some of the harder cancers commercially we can sort of add on. And ultimately, there's a lot of shared signatures between cancers and a lot of different ones, and leveraging these different signatures and then sort of attacking them one at a time, so that ultimately we don't have to wait. Right now, our best way to detect cancer, most of the cancers, is wait till we get symptoms, right? Except for screening like for colon cancer, or PSA, which is controversial. We have mammography for breast, and then low-dose CT for lung. But again, in very select populations. So, but most of the time we are waiting for symptoms and people to get sick. So how do we get in front of that? And so that's what we're doing. And I think all the work that's coming from the different genomics efforts, and we also use proteomics and all these looking at transcriptomics is now showing us the signature. Then now we can then with AI to figure out what combination of those signature is going to be most cost effective, most sensitive and specific, as everybody's cancer is different, to understand the heterogeneity so that we can detect all these cancers while they are very easy to deal with. Most cancers, if you catch them in stage 1 or 2, completely curable. So if we're able to shift even up a stage, we'll be saving easily millions and millions of lives.
And are you screening based on like history, or like are you incorporating genetics into deciding who you're going to screen, or how does that piece work?
Yeah, our test is designed for a healthy population, because right now, if you're over ages, depending on which country you're in, 45 or 50,
right.
you are at a higher risk, and governments now pay for screening for even if you have any symptoms or even family history. But colonoscopy is very invasive, and some countries, and then stool testing people don't want to do, and a lot of people are still not getting screened. So we're providing this alternative blood test for people who are not at elevated risk, who maybe just because of their age, at a higher risk, and to be able to screen these populations.
Well, Kelly, we talked about organoids, right? A little bit.
I did. Yeah. That would have been my third area.
Yeah. I think this kind of links to precision medicine also. I just want you to, like talk about your interest.
Yeah. So, in thinking about like as a research institution, where do we want to put money? I think I would put a plug in for developmental biology, because it's really only through developmental biology that we've been able to build the models that for disease, and also for testing drug efficacy and safety in human systems. So obviously you can't test it, or you don't want to test it on a human being, but we're now starting to be able to create like body on a chip, human organoids, fused brain organoids, for example. And we've only been able to do that because we understand how these cell types and how these tissues form through development. But we're not there yet. And a lot of the cell types that are getting produced are still quite immature, and we're not seeing the full breadth of tissue heterogeneity, for example. We're not seeing the full breadth of responses, metabolic responses, whatever. And so there's still a long ways to go. But I think these technologies that are emerging to test drugs and test efficacy and understand disease in human systems, whether it's human organoids, human on a chip, humanized animals, whatever it is. I think these are really, really powerful technologies as well that are coming out. So nothing to do with genetics per se, other than understanding how these systems form during development. That's something I'm really excited about, and actually something that is close to my heart, because it's much closer to my background in stem cells and developmental biology. So I am biased, but yeah.
Oh, we're all biased by our background. No, so I think I must be doing a great job, because no questions. So we're just gonna, we've been at this for 55 minutes, and I think we're gonna now, I'm gonna ask first Jimmy now, giving him heads up. Jimmy, I mean, where do you think? And I know I know it's a hokey question, but in the near future, what do you think? Like, from your field, that this is not possible right now, but I do think this is gonna be possible in a couple of years. Right now, right now. From the forefront, right now we're still not there, but you know what? In 3-5 years, I think this is gonna benefit a lot of people. So those kind of a specific view of what's gonna happen.
Yeah, so my field is oncology. So I think we've been talking mostly diagnostics, but I think there's a combination of multiple things, right? In oncology, let's talk about therapeutics, where in the past, our most of our drugs is just sort of toxins, and we hope to, like mustard gas derivatives. So we hope to kill the cancer more than the person, and chemotherapy, right? But now we're having whole new paradigms of new ways of treatment, whether it's immunotherapy, cell-based therapy, whether it's oncolytic viruses. So there's an explosion of entire new modalities of treatment there that we're thinking about, and then leveraging our own immune system, right? The last few sort of Nobel Prizes is as much chemicals that we can give, sometimes using our own immune system is very is the best way. So I think so there's advancements in the therapeutics aspects, and then combined with advancements in diagnostics of some of the work that I'm hoping to contribute as well. I do think that cancer could be a chronic disease in our generation. Many of the cancer types that we know now that are very life limiting can be either detected early or even if detected later with the right approaches of therapeutics. I think that we are at the beginning of the end of cancer, and I'm pretty optimistic there.
Yeah, I really realize that precision medicine is not a one shot, one kill. I think I realize that precision medicine really means that there are a lot of options that might fit you, and finding that right option is the basis of precision medicine. It's not a one size fits all. But like I said, from oncoviruses to everything right now, just having that large, arsenal, should I say, is just definitely very, very hopeful for. And being a cancer patient family, is those are the things that really bring hope, rather than just one size fits all. So definitely encouraged to hear that that we're definitely explosiveness of these new oncoviruses and immunotherapies and Optivo, right? And everything right now. I think PD-1, and I think I hope that we hope to see more. And the key thing is that quality of life, and I'm very happy to hear that you're all optimistic also that we're that moving moving that way. Caroline, and then you had a head start now with Jimmy.
Well, no, it's not a head start because I was listening eagerly to Jimmy and I wasn't thinking about my answer. No, I mean, I think one of the things that we're already starting to see and will come out the better the data that we generate in the future is really this idea of preventative medicine and being able to stratify risk in populations and to tailor treatments to people that not treatments, tailor interventions to people that need them the most. So, being able to pick up this. So this is based on genome sequencing. So I can imagine, in some kind of utopian society where everybody trusts the government, that you could go and have your blood tested and have your genetics on file, and basically have a system of understanding what you're at risk for, based on the intersection of your genes as well as your environment, and to start to have early interventions. We already have that in a sense. Like, we have this idea of risk, for example, for breast cancer is family history and mutations in BRCA and other genes and whatever, but there are lots of other diseases where that similar kind of rationale would apply, where if you fall into a certain risk category, then you really need to be having, screening more frequently at an earlier age and trying to mitigate the risk, basically, to pick it up earlier and to deal with it. And so I think that's something that we'll see coming out in some societies. I think it will be harder in some places like the US, where there isn't so much trust and there isn't such a well-integrated medical system. I know, like, Australia, for example, has a really good well-integrated medical system, electronic health records, and a single sort of government provider in most cases. UK is the same, and both of these populations have a really high level of trust in their governments. And I think that's very, very different to what we see in the US. And that's going to be much more sort of state by state and not necessarily accessible to all people as well. So that's kind of another hurdle, is the ethics of it and the socioeconomic implications of it as well. But I think on the whole, this idea of preventative medicine, genomic health, as opposed to disease is an idea that I'm really excited about, and I think it is something that we'll start to see a lot more of.
Well, I don't know you guys, but it really flew by for me, because having two of you guys really, once again, Jimmy from logging in from Taipei, Caroline driving through a tornado to make sure you get a connection to I'm sure a lot of people around the world, right now who's logged in, I hope they found it was it was beneficial. But I definitely think that we can come away with that we are making progress, and all of us are trying to make things make things better, and I hope to see you, Jimmy, soon, and Caroline also. And
Yeah,
then,
absolutely.
and then thank you for everybody who logged in on the webinar. Thank you very much.
Great. See you later.
You there. Thank you.
Good to see
Bye
you.
bye.